We usually treat vitamins like generic health boosters, something you take in the morning to feel a little more energetic. But biologically, a vitamin is a chemical vulnerability. It is a highly specific compound that the human body needs to stay alive, but lacks the internal machinery to build from scratch. Because we cannot manufacture these molecules ourselves, our biology is trapped in a permanent state of tension. We are entirely dependent on the outside world, forced to constantly extract these chemicals from our environment to survive. These essential compounds are not scattered randomly across the food web. They are hidden in very specific locations, locked away in plant and animal tissues by the strict laws of molecular chemistry. To track down all 13 essential vitamins, we have to look at this catalog as two distinct biological teams, separated entirely by how they dissolve. One team is soluble in a lipid droplet, meaning fat. The other team is soluble in a water droplet. A vitamin's chemical solubility dictates the search. It reveals exactly where that nutrient hides in the environment and how our bodies must manage the supply. Let's open the first volume, the fat-soluble vitamins, which include A, D, E, and K. Because these molecules only dissolve in lipids, they are exclusively stored in the fat-heavy tissues of plants, animals, and our own bodies. Vitamin A, for instance, exists in its primary form as preformed retinol. If you want it, you have to look inside the fat reserves of animal sources. It's heavily concentrated in liver, whole milk dairy, and egg yolks. But herbivores have a workaround. Plants produce an alter ego called pro-vitamin carotenoids. Humans can extract these precursors from dark green leafy vegetables or bright yellow and orange plants and convert them into retinol during digestion. Then there is vitamin D, the biological oddball of the catalog. While we classify it as a vitamin, it functions within our systems as a pro-hormone. This diagram shows why. While we extract a fraction of vitamin D from fish, we synthesize 90% of it directly in our skin. When UVB radiation penetrates the epidermis, it physically strikes a cholesterol precursor molecule, converting it into active vitamin D3. Because vitamins A, D, E, and K are fat-soluble, the human liver can pack them away in its own lipid droplets for months at a time. It's a biological savings account that prevents immediate starvation if a hunt fails or winter freezes the local vegetation. Vitamin E acts as a microscopic bodyguard, protecting polyunsaturated fatty acids from oxidative destruction. To find it, you have to break into fat-dense botanical vaults, vegetable oils, hard seeds, and nuts. Vitamin K operates with a split identity. The first form, K1, is structurally bound to the chloroplast membranes of dark leafy greens. But the second form, K2, has a radically different origin. You don't forage it from the outside world. This map shows the human colon, where a symbiotic colony of anaerobic bacteria synthesizes K2 entirely in the dark, right inside our gut. If we do get K2 from the outside, it comes exclusively from fermented foods, where those exact same bacterial cultures thrive. To acquire just these 4 fat-soluble nutrients, early humans had to hunt animals, press oils from seeds, eat leaves, and outsource their own metabolism to a bacterial microbiome. The second team operates under a much stricter rule. Water-soluble vitamins dissolve in water, which means human tissue cannot easily store them. Because our blood and urine are water-based, the body is constantly flushing these nutrients out. This creates a relentless biological ticking clock that forces us to secure a daily dietary resupply. This timeline charts the evolutionary backstory of vitamin C. Unlike most mammals, human ancestors suffered a specific genetic mutation that destroyed the L-gluconolacto-lactose oxidase enzyme pathway. That single mutation severed our ability to synthesize vitamin C from glucose. Now we are locked into a dietary dependency. To get it, we have to consume water-rich, fresh vegetation, citrus fruits, bell peppers, and tomatoes. The inability to store water-soluble vitamins meant early humans could never just eat a massive meal and hibernate. We were forced to become continuous, daily foragers of fresh plants just to keep our cells functioning and stave off scurvy. That brings us to the B complex, an 8-part catalog of essential metabolic coenzymes that are wildly scattered across the entire food web. B1, thiamine, whole grains and pork. B2, riboflavin, dairy products and eggs. B3, meat, fish, and intestinal bacteria. B5, inside mushrooms and avocados. B6, inside bananas and poultry. B7, egg yolks and seeds. B9, folate, spinach and legumes. But then we hit a strict biological outlier, B12. Look at this grid. While the other 7 connect to a wide array of plants, fungi, and animals, B12's connection lines isolated completely. It is the only water-soluble vitamin absent from the plant kingdom, synthesized strictly by bacteria and found exclusively in animal products. Beating the ticking clock of water solubility and avoiding a B vitamin deficiency requires cross-referencing and consuming nearly every distinct food group on Earth. Look at the completed map. To keep our cells running, we have to extract chemical compounds from plant chloroplasts, animal livers, seed oils, the bacteria living in our colons, and the radiation of the sun. This massively scattered chemical catalog proves exactly what kind of animal we are. We are obligate generalists, forced by our own enzymatic limitations to interact with the entire surrounding ecosystem. It also exposes the modern marketing myth of the superfood. No single ingredient in nature, no berry, grain, or meat, contains the complete chemical profile required to sustain human biochemistry. Our chemical dependency on the natural world demands a wildly diverse diet, a blueprint of survival that connects our internal biology to the entire ecosystem.